Gene and genome. Gene. Gene is information stored in DNA, and that information is eventually the information to make a protein. And in the intermediate state, the DNA is copied into mRNA through transcription, and then the information is translated into protein and utilizing ribosome. The gene structure is a little complicated. The gene on the DNA, it contains exons and introns. The information which describes the protein structure is only in exons. So those exons are separated by intervening introns. So when mRNA was made, both of those exon and intron is transcribed, and this mRNA is a pre-mRNA. And after all the introns are removed, then mature mRNA is exported to the cytosol. Now this mature mRNA sequence is real genetic code which describe the structure of proteins. Then what is a genome? Genome is the collection of all the genes, and then those genes are spread out the whole chromosome or whole DNA. Human Genome Project is the project which sequence whole human genome, which is composed of 3 billion base pairs. This project was led by the Human Genome Project team. This is an internationally organized team, mainly led by the international governments. Some years later, another small venture company, Celera, led by Craig Venter, joined in this competition. Two teams utilized the two different approaches. Human Genome Project team and break down those genome into relatively big fragment first, and each fragment was further fragmented into smaller pieces. Then those smaller pieces were sequenced one by one, then aligned together. And this process provides the reference genome. And then this genome or the sequence can be used for next step. Celera team, they skipped the first step and they fragmented those genome into very small fragments from the beginning and then tried the shotgun approach. After that, they used this small fragment to align to make the full genome sequence. Looks like this is a quite fast approach, but later it was found that they used this reference genome to match or to map these fragmentary informations. Human Genome Project started in 1990s, and the original plan was for 10 years. That's why in 2000, so it was kind of politically announced that the Human Genome Project is almost done, and one team is the Human Genome Project team, and the other person is the Craig Venter, the Celera team. And the next year, they published two papers separately. Celera team published the result in Science, and Human Genome Project team published their result in Nature. The first draft was published in 2001 and formally declared the completion of the project in 2 years. And the result covers the human genome 3 billion base pairs. But this result is only covers euchromatic region, that's 90% of the total genome. And that excludes the heterochromatic area, such as the centrosome or telomere. The result of this sequence show that our gene number, the information to make proteins, that means our protein number is just above 20,000. This is a surprisingly lower number than what we expected before. Because human is so complicated, we believed we may have a lot more numbers of proteins. That's why this number is surprisingly smaller than we expected. And also interestingly, this protein coding area of the gene is only covers 2% of total DNA. Then what is the other 98% of DNA? That's a natural question, isn't it? In 2007, the first individual genome was reported. Can you guess who that was? The classical definition of a gene is the information to make a protein. But very interestingly, after Human Genome Project is done, only 2% of genome sequence is related to this information. Then how about the other 98%? Well, maybe half of those DNA information, they are transcribed into RNA, but they do not make a protein. So that's why this RNA is called as non-coding RNAs. So now, new function of these RNAs are the topic of genetic studies. Then what are the examples of non-coding RNAs? Well, ribosomal RNA, they have a function and they are composed of RNA molecules. And these are one example, very typical classical example of a non-coding RNA. Also tRNAs. This is also important for protein synthesis. And microRNA, siRNA are the other very important, the functional RNA molecules. Now, after this, the Human Genome Project, now we have the term of a genome, the total collection of gene, and then we have transcript, and all together, this transcript together we call transcriptome, and protein, and all collection of a protein we call proteome, and glycan structures, all the carbohydrate together, glycome, and metabolite, all collection we call metabolome, kinase, kinome, phenotype, phenome, kind of different words is kind of spreading out at this moment. So this new post-genome era, we have a lot more information to handle the total set of informations to study and to understand the complicated biological systems.